right, we're going to start talking about vaccines. Currently, it's February 2021. We have just recently gotten our first COVID-19 vaccines out. Those are relatively new types of vaccines, and there's other types of vaccines that haven't come out yet. So we'll be having a specific separate lecture on COVID, as well as a specific lecture on influenza. But here we're going to talk about vaccines as a whole. So this explanation might make an immunologist cry, and I apologize to you if you're an immunologist. But for the sake of understanding how vaccinations work, we really only need this basic understanding of how the body is going to respond to an antigen. And so an antigen is going to be usually a protein that is able to be seen and responded to by the immune system. So in this case, we have a disease-causing virus. It's got an antigen on the outside of it. Usually the antigens that we're going to use in vaccinations are going to be on the exterior of the virus because that's where the body's immune system is going to be able to recognize it and see it most easily. So we're looking at this antigen on the outside of this virus, and the body doesn't have an immune response ready yet. So that virus is going to enter the body, it's going to replicate inside cells. At a certain point, the body's immune system comes in, it recognizes that there's a problem, it starts to kind of non-selectively attack those cells, and then it presents pieces of the virus, those small pieces that are recognizable, those antigens, and presents them to different cell types in the immune system. One of them are the helper cells that are then going to go and activate a couple of different types of immunity. So they're going to activate cell-mediated, which we're really just thinking of as T-cell immunity, and they're going to activate antibody-mediated, which we're looking at as B-cell immunity. So it's the B cells that are going to produce antibodies, and it takes a while for them to do that. So it's going to take 2 or more weeks in order for the body to produce antibodies against that antigen. The cytotoxic or killer T cells are also being activated against that antigen. They don't make antibodies, but what they do is they release cytokines. And cytokines are those small molecules that can cause inflammation. They're there to help attack infected cells, and they can act directly on cytokine receptors in our cells. They can promote free radicals. They can cause cell death in many ways, but they're not specific only to the cell that is infected. So when you have this cytokine release, you're basically attacking any cell in that immediate vicinity. So when we talk about how vaccinations work, what you're coming away with is you're going to have a virus that has an antigen on it. Any way that we can replicate that antigen or present that antigen into the immune system is going to cause activation of the cell-mediated or T-cell-mediated immunity as well as the B-cell-mediated immunity. That's all based on whatever antigen we gave it in the vaccination. You've got that antibody-based, which is going to take at least a couple weeks, and we've got that T-cell pathway, which can release cytokines and can help attack the infection, but it can also have some side effects of attacking us if it goes too far. Vaccines generally actually activate both B and T cell pathways. Some vaccines are going to activate mainly the B cell part. Some of them are going to have more T cell activation. So there are several different types of vaccines, and I want you to watch these videos. There's a really good explanation of the viral vector and the mRNA vaccines, but I'm going to explain them as well. So remember, the whole goal of the vaccine was to present the antigen to your body. And in terms of creating an immune response, the body doesn't care how the antigen gets there. It just needs to see it. So we've developed multiple ways to get that antigen there. The original way, which was kind of the most obvious way, was to grow that virus as a whole virus and then either inactivate it or kill it so that it could no longer infect humans. So let's just get rid of that. But that it kept the antigen on the outside of it. The immune system sees that, it attacks that antigen, and that's your immune response. You can also use a whole cell that hasn't actually been completely inactivated or completely killed, but is just very weakened so that it's unlikely to produce any infection in the human body. That's still going to look like this in that you're going to have the antigen on the outside of the cell looking just like it normally would. With the inactivated whole, there's absolutely no way to be infected with that virus. With the live attenuated, there is a very small chance of being infected, and it usually would only occur if someone has a very weakened immune system already. If you don't want to give the whole virus, you could go the route of just giving that antigen protein. So there's a few ways of doing that. You could grow the whole virus just like you did previously, and then purify out that antigen protein and throw away the rest of the virus. You could use genetic engineering, and you can grow just the one section of that protein that you want to make your antibody against. So that would be a recombinant antigen protein. Or you could produce a virus-like particle, which just has a very similar cell envelope as the virus and contain the antigens on it. In terms of effectiveness, the more the antigen actually looks like it could be coming from the virus, the more effective the vaccination is going to be. So a live attenuated is going to be very effective, and for the protein ones, the virus-like particle is going to be more effective than the purified or the recombinant because it's in the shape and form of that surface protein. Because those single protein antigens are less effective, in order to use these for vaccines, you often have to give what we call an adjuvant, which is a chemical that additionally activates the immune system so that it's more responsive to that antigen protein. So with these, you're often going to see an adjuvant along with it. These are great. Everything in 1 and 2 are basically giving your body that protein in very different forms, but it's giving the body the antigen protein itself. The next 2 that we're going to look at aren't giving the protein, they're giving the nucleic acid, genomic information for the protein, and they're counting on our cells to create the protein and express the protein ourselves. So the idea here is, of course, you don't want to give the full genome of the infectious disease because you don't want to infect someone. So you just take the genomic code for that one antigen protein. You can give it either as mRNA or as DNA. And if you give it as mRNA, that has to be absorbed into host cells, which are then going to make proteins of the antigen, stick that protein onto the outside of our cell, where then it's available for the immune system to see. Same thing with DNA vaccines. That's going to be absorbed into the host cells. We're going to make mRNA from it. Then we're going to make protein and express that protein on the outside of our cells. The difference between the RNA and the DNA vaccines is that RNA is very labile. It's fragile. It can break down very, very easily. DNA in this circular form is not nearly as fragile, so it's easier to store, easier to transport, and can be kept longer. So that's giving the nucleic acids alone. In both of those cases, you're counting on the cell absorbing that nucleic acid. And those vaccines seem to work fine, so we know that cells do that. But if you were trying to design a more effective way of getting that nucleic acid inside the cells, you could say, I don't really want to wait for it to just be absorbed. I want to directly inject that nucleic acid inside the host cell. And that's what they're doing with these vector-based. So vector-based, also called viral vector, means they're actually using a second virus. So they're using a virus from a common cold or some type of virus that isn't very dangerous for us. And they're replacing important parts of its genome with the gene of the antigen. So the viral vector shouldn't be replicating, but all it's doing is injecting the antigen gene into your cells. That's very efficient because it gets that gene exactly where it needs to go very quickly. But there are some problems with it in that once your body has seen that vector virus, it's going to have an immune response to the vector as well. So some of the concerns about using these viral vectors are you might make a vaccine with one viral subtype as its vector, give that to everybody, and then the next time you need to make a different vaccine, you can't use that vector anymore. You have to find a new one because now all the people who were vaccinated to begin with are also immune to the vector. So there's pros and cons for all of these. So just to summarize those, you had the whole virus. You could either inactivate it or you could weaken it, live attenuated. Influenza vaccines can be either. The most common are the inactivated form. And there are different ways to inactivate them, but often that's done by heat. The inactivated form is very effective. However, the live attenuated or weakened is more immunogenic than the inactivated, so it can create a bigger immune response, which can be good, but if you have a very poor immune system, there is always that possibility that you could become infected. One important thing to know about how these are being grown or synthesized is that these viruses have to be grown the way a virus does. It has to be inserted into some type of cell, which then replicates the virus because it's infected. When we produce these vaccinations, we industrialize the cells that we're going to infect and industrialize, purifying the resulting virus out of those cells. So influenza A is often grown in chicken eggs. Chicken eggs will replicate it very quickly and efficiently, and then the virus is purified out and then inactivated. However, with the purification, you can't guarantee that 100% of the chicken egg proteins are left behind. So this is why when people get an influenza vaccine, they're asked if they have an egg allergy, because if they do, they could potentially have an allergic reaction to peptides or microparticles from those eggs that it was grown in. Most of the live attenuated or weakened viruses that we use for vaccinations are not grown in eggs. Most of them are grown in cell cultures that are grown in vats, and those cell cultures could either be yeast or insect cells or mammalian cell lines. So those protein-alone vaccines, we talked about purifying antigen proteins. Those are also called subunit vaccines. And you're going to make those the same way as you made the whole, and then you're just going to purify out only that antigen protein. We also have recombinant antigen protein, and that's grown inside mammalian, yeast, or insect cell lines and purified out. Again, remember, these purified antigen proteins aren't super strong in terms of immunogenicity on their own, so those are the ones we usually will end up having to add an adjuvant. They'll also usually require a booster, but you're only having your immune system react to one protein and not to a large number of them, so they can have less side effects than a whole virus vaccine. The vector-based vaccines that we're looking at now use an adenovirus as the shell, so you'll often hear these referred to as adenovirus vaccines or adenoviral vector vaccines. And that's just referring to the shell virus that the vaccination gene is being inserted into. As I said, these are very immunogenic because they're actually inserting the gene inside the host cell. But again, you may only be able to use each vector once. So in terms of design and manufacturing of these vaccines, that produces some limitations. We're going to talk about COVID vaccines in more detail in future lectures. But I do want you to recognize that the Johnson & Johnson and the AstraZeneca Oxford vaccines are vector-based. They're in adenovirus vectors, and these are produced in human cell lines, and we'll talk about that a little bit more. And then you had those nucleic acid, whether it was mRNA or DNA. And for COVID, the Moderna and the Pfizer vaccines are both mRNA vaccines. You've almost certainly heard about the problems they're having with these first COVID vaccines, in that they have to be kept at extremely cold temperatures. They have to be used within a few hours after opening, and that's all due to the fact that mRNA is very, very fragile or very labile. The DNA ones that they're developing are going to be able to have easier storage and longer lifespans. As far as how they're growing or synthesizing them, these are the only ones that don't need to be produced in a cell line of any kind. Because it's just pure RNA or DNA, they're chemically synthesized, no cell lines of any sort. So some vaccines need boosters. There are some shots that are only one shot, it lasts your whole life. There are other shots that are a 2-shot regimen, in which we call the second shot the booster. And what that's really coming from is how long those memory cells last after you got your first shot. So if you're getting your primary vaccination, you're waiting a couple of weeks, you're going to make your antibodies. Those are going to go down after a while if you haven't been exposed again. But you're also making those memory cells, and they're going to last longer. Depending on the antibodies that you make, which depend on the virus and depend on the vaccine, those memory cells are going to decrease. And once they decrease enough, then the vaccine's not going to work anymore. So we try to go in at a certain point before those memory cells go down too far and give a booster vaccination, which then reactivates that system. Sometimes we only need to do that after several years, like tetanus shots. Sometimes we need to do 2 in a row immediately in order to get an acceptable level of immunity, such as these first COVID shots. So these boosters are the same shot both times. We haven't changed the type of vaccine in that shot. So it's not the same as giving updated vaccines. So with influenza vaccines, we're not giving boosters, we're giving new ones, but we're giving a new one every year. And that's because the flu itself is mutated and we needed a new vaccine. So boosters are the same vaccine, but given multiple times. And the updated vaccines are when there's been a mutation and you need a new vaccine. So that idea of mutation and vaccinations is very problematic because when you think about the vaccination, you're creating antibodies and T cell responses to a specific antigen. And that antigen is being recognized based on the shape and the charge of it. If there's a mutation in the virus that changes the shape and charge of that antigen, then it may or may not be able to be recognized by your immune system. And we call that antigenic drift, when mutations can cause the shape of the antigen to change over time, and this can end up reducing the vaccine effectiveness. So how much antigenic drift you get depends on how fast that particular virus mutates. And there's a great diagram here from the Journal of Virology, and on the y-axis is mutation rate, so we're just going to say this is a high mutation rate, and this is a low mutation rate. And they've got it by genome size, but I'm just going to show you that we've got bacteria down here. Bacteria have a higher mutation rate than humans do because they don't have as good a proofreading. But viruses up here have even higher mutation rates. And it's not 100%, but you can see that there's a trend where these DNA viruses down here have a lower viral mutation rate. And the RNA viruses, including HIV, tend to have a higher mutation rate. So theoretically, these high mutation rate viral types, these RNA viruses, are going to have faster antigenic drift and are more likely to require updated vaccines. And when you look at some of the examples here, we see in these high viral mutation rates, we see influenza, which we know we do make new vaccines for every year. We see HIV and Hep C, which despite all the work immunologists have been doing, we still don't have effective vaccines for. And we see coronaviruses up here. And it's the fact that the coronaviruses have this very high mutation rate that's similar to influenzas that lead us to believe that it's likely that there'll be new strains every year and that they'll need to have new vaccines every year, just like we need new vaccines for the influenzas. So you guys are going to be going out in clinic this summer. You've probably already volunteered at vaccination clinics. And if you haven't, please go out and do so. And you're going to have people with some basic concerns about vaccination. I'd recommend that you pull this up and take a quick look at it before you're involved in any vaccination events or interacting with any patients that'll have questions about it. There are a few concerns that are very common for people to have about vaccinations. So I wanted to just go over those very briefly so that you're ready when you go out there to answer these. The first one that's being asked is about an ingredient in vaccines called thimerosal. And thimerosal, I'll just start by saying it is rarely used anymore. It's a mercury-based antiseptic and was put into vials in order to keep bacteria or fungi from growing and to keep the vaccination from breaking down. The most common use was in multi-use vaccine vials because no matter how aseptic you are, every time you go in there with the needle to pull another dose, there's some chance of introducing bacteria or fungi. So they just wanted to have some form of antiseptic in there. I'm not going to go into the chemistry of it. There's literature out there that you can look at. But what your patients may want to know is that there are no childhood vaccines in the US that have thimerosal in them. So it was back in 2001 that thimerosal was removed from all childhood vaccines. So none of those vaccines that kids are getting in their standard vaccination protocols will have it. The only vaccine they may be exposed to that has thimerosal is that from multi-dose vaccine vials for influenza. However, there are other influenza vaccines that are packaged differently that do not have thimerosal. So if you do have a patient who is concerned about that, they can definitely be given a thimerosal-free version. The second concern that I have to mention is a concern that's out there about whether autism can be either given or exacerbated by vaccination. And I cannot go into this topic. I have taught an entire course on autism, and I could discuss evidence-based information on vaccines for over an hour, but I won't. What you need to know as adult healthcare providers who are interested in evidence-based medicine and have the ability to go out there and look at evidence in the literature is that there is no known connection between autism spectrum disorder and vaccinations. There's plenty of information out there on the difference between correlation and causation, changes in autism diagnosis, studies that have been done using current diagnostic criteria for children, adults, and elderly that find if you use the same criterion, they have the same rates of autism. Multiple different forms of evidence. But the end story to it is not only is there no known connection, but there's actually evidence that there isn't a connection. The last concern that I've heard put out there recently about vaccination, especially in terms of COVID vaccination, is whether there are fetal cells used to produce these vaccines. And for some of the vaccine types, the adenovirus types, that's a little bit of a murky answer. So let me explain that. So we know that these vaccines are grown inside of eggs or inside of cell lines, and then they're purified out. The protein vaccines, the live inactivated vaccines, those are grown inside eggs or insect cells or yeast or other mammalian cells that don't have anything to do with embryonic tissue. Adenovirus vaccines, however, are grown in cell lines that were derived from human fetal tissue. One of the things I've seen out there are people saying that the vaccines themselves contain embryonic tissue or fetal tissue, and that needs to be clear that no vaccine contains any human cells or tissues. It is true that the adenovirus vaccines are grown in a cell line that is derived from fetal tissue. So there are 4 vaccine-related cell lines that have been created from aborted fetal tissue, 2 of them in the 1960s, one in 1972, and one by Johnson & Johnson in 1985. These are the only cell lines derived from fetal tissue that are used in any vaccine process, whether it is the basic science design or the manufacturing. So it's important to know that there's no new fetal tissue being used, no new cell lines are being created, and no new tissue is being used for production. That said, it is true that the adenovirus vaccines are grown in these cell lines. At the moment, specifically the COVID, AstraZeneca, and Johnson & Johnson vaccines. So if you do have a patient who does not want to have one of these vaccines, there are other vaccine types available for them. And if they're concerned about a vaccine that's grown in any type of cell line, it's important to know that RNA, DNA vaccines are not using cell lines of any kind. They're synthesized chemically in a vat. So hopefully that clarifies some information for you. And again, this lecture is introductory and is supposed to be an overview of viruses in general. We're going to have future lectures that are going to talk about influenza virus and vaccines, COVID virus and vaccines, etc. So if this lecture made you motivated to know more about those vaccines, then hopefully you'll come and listen to my next lectures.